Hello viewers, welcome to the biotech path. Today we are going to discuss a new mode of treatment to cancer, what is called synthetic lethality. This concept evolved long time back but came into the clinic only 10 years back. Let us discuss the details. Let us say there are two genes, gene X and gene Y in a cell. When gene X is lost, the cells survive. When gene Y is lost, the cells survive. But when both the genes are lost, the cells die. This concept observed in certain organisms was called synthetic lethality. Let us look into the history. The initial concept was discovered somewhere in 1922. It was observed by Calvin Bridges. He first observed in the fruit fly Drosophila melanogaster, where he found specific combinations of mutations led to death, while individual mutations did not affect the viability. The term synthetic lethality was later coined by another scientist, Theodosius Dobzhansky, in 1946. However, the application to cancer did not materialize till 1997 when Leland, Geoffrey Friend and colleagues proposed that this synthetic lethality concept can be used to treat cancers. Okay, in cancer cells, how we can use this? Like I told you, instead of gene X or gene Y, I put it as gene 1 and 2 here. Let us say there are two genes, gene 1 and gene 2. Cancer cells, have mutations. So let us say a cancer cell has a mutation in gene 1, still the cancer cell will survive. Let us say a cancer cell has mutation only in gene 2, then also the cancer cell will survive. If these two genes happen to be synthetic lethality genes, then if both the genes are knocked down by some mechanisms or by mutation, then the cell will die. So we have to choose a therapy which will either knock down gene 1 in case the cells are mutated in gene 2. If the cells are mutated in gene 1, we will choose an inhibitor that will knock down gene 2. In this manner, the cancer can be treated. Now, the practical example being the discovery of what is called PARP BRCA interaction. You might have heard about this. This came into popularity somewhere in 2005 when BRCA1 mutations were discovered. BRCA1, breast cancer gene. Thomas Ashworth and Halday demonstrated a synthetic lethal relationship between these two genes. One is you can inhibit PARP, or cancer cells, as you know, are often mutated, especially the breast cancer cells and the ovarian cancer cells are mutated in BRCA1 or BRCA2 genes. The first clinical approval came in 2014 when the PARP inhibitor, what is called Lynparza, was the first synthetic lethal drug which was approved by FDA for treating advanced BRCA mutated ovarian cancers. Now in breast cancer, there is a type of breast cancer which you might have heard. It's called triple negative breast cancer. It's somewhat difficult to treat. I'll tell you why. Because this particular cancer, why it is called a triple negative? Because the usual receptors that are present, such as estrogen receptor, progesterone receptor and HER2 receptor, they are absent in this particular form of breast cancer. So what happens is, we have medicines for down regulating or targeting these receptors. These receptors can be used as a target, but then since this cancer has no such receptors, it is difficult to find a suitable target. So lack of targets kind of limit the treatment options other than the conventional surgery and chemotherapy. So we need to have some kind of novel treatment for these kind of cancers. That is where this synthetic lethal concept helps. For example, breast cancer gene 1 or 2 is usually mutated in breast cancers. What happens is, these genes are also involved in DNA repair mechanisms. So they cannot fix DNA breaks properly if these genes are mutated. PARP is another gene which is also involved in DNA repair. So if you inhibit PARP, then the cells cannot fix DNA breaks. So this is what happens if you have a combination of these two. Breast cancer cells, they already have BRCA1 mutation, and now if you give PARP1 inhibitors, then the cancer cells will die off, sparing the normal one. That is the importance. There is another importance to this. PARP1 inhibitors are already in the clinic, and so this is sometimes called, if you are using such drugs which are already in the market to treat cancer or any other disease, then you call this as repurposing of drugs. They may be used to treat some other forms of diseases, but if you use it to these kind of disorders, then you call it as repurposing of drugs. That is one thing. Second thing, the BRCA1 mutation is found only in cancer cells. So this drug is now going to affect only those cells, leaving the normal cells intact. So this is called precision medicine. So in that sense also it is very useful. Now after that success of Olaparib, which I told you, it has been approved in ovarian cancer, breast cancer, pancreatic cancer and prostate cancers. Similarly, there is another PARP inhibitor which is called Repurposing. It is also approved for treating ovarian cancer and prostate cancers. There is another one, Niraparib, which is approved for ovarian cancer, fallopian tube or peritoneal cancers. Now we need to talk about other synthetic lethal strategies. For example, MYC and PARP inhibition. This is also used to treat triple negative breast cancer cells. They are also looking into EGF receptor and PARP inhibition, particularly again in certain types of triple negative breast cancers. Similarly, combination of with other drugs is also being attempted, like topoisomerase inhibitors, to improve the outcomes for both BRCA1 mutated and non-mutated triple negative breast cancers. Now, so far we have described what is synthetic lethality and what are the ones that are already in the clinic, and how there are many similar treatment modes emerging. So, why this is important? One of the most important things that you might have noticed is that we are trying to make use of the weakness of cancer cells. That is an important thing, to find new targets. And since the mutations are restricted to cancer cells, this is one way to minimize damage to the healthy cells. And last but not the least, it opens doors for personalized therapies. So we can pay attention to individual patients and their defects and design the treatment accordingly. Thank you for watching.